increasingly by the use of chemical by use of insecticides and pesticides. These chemicals are toxic and extremely harmful to human beings and animals alike and have been polluting our environment, soil, water, foods, vegetable and crop plants. Our soil is also polluted through our use of pesticides to remove weeds, biological control of pests and disease. In agriculture, there is a method of controlling pests that relies on natural production rather than introduced chemical. A key belief of the organic farmer is that biodiversity furthers health. The more variety a landscape has, the more sustainable it is. The genetic farmer, therefore, was to create a system where the insects that are sometimes called pests are not eradicated but instead are kept at manageable level by a complex system of check and balance within a living and vibrant ecosystem. Contrary to the conventional farming practices, which often use chemical methods to kill both useful and harmful life forms indiscriminately, this is a holistic approach that seeks to develop an understanding of the webs of interaction between the myriad of organisms that constitute the field fauna and flora. The organic farmer holds the view that eradication of the creatures that are often described as pests is not possible but also undesirable, for without them the beneficial predatory and parasitory insects which depend upon them as food or host would not be able to survive. Through the use of biocontrol measures will greatly reduce our dependence on toxic chemicals and pesticides. An important part of the biological farming approach is to become familiar with the various life forms that inhabit the field, predator as well as pests, and also their life cycle pattern of feeding and habitat that they refer. This will help develop appropriate means of biocontrol. The very familiar beetle with red and black markings, the ladybird and dragonflies are useful to get rid of aphids and mosquitoes respectively. An example of biological control agent that can be introduced in order to control butterfly caterpillar is the bacteria Bacillus thuringiensis, often written as Bt. These are available in sacs as dried spores which are mixed with water and spread into vulnerable plants such as brassicas and fruit trees where these are eaten by the insect larva. In the gut of the larva, the toxin is released and the larva get killed. The bacteria disease will kill the caterpillar but leave other insects unharmed because of the development method of genetic engineering in the last decade or so. The scientists have introduced B. thuringiensis toxin genes into plants. Such plants are resistant to attack by insect pests. Beauty cotton is one such example, which is being cultivated by in some states in our country. A biological control being developed for use in the treatment of plant disease is the fungus Trichoderma. Trichoderma species are the free-living fungi that are very common in the root ecosystem. They are effective biocontrol agents of several plant pathogens. Baculoviruses are pathogens that attack insects and other arthropods. The majority of baculovirus used as biocontrol agents in the genus Nucleopolyhedrovirus. These viruses are excellent and for species-specific spectrum insecticide application. They have been shown to have no negative impacts on plants, mammals, birds, fish, or even on non-target insects. This is especially desirable when beneficial insects are being conserved to aid in the overall integrated pest management program or when an ecologically sensitive area is being treated. Microbes as biofertilizers. Within our present day lifestyle, environment pollution is a major cause of concern. The use of chemical fertilizer to meet the ever increasing demand on agriculture produce has introduced significantly to this pollution. Of course, we have now realized that there are problems associated with the overuse of chemical fertilizers, and there is a large pressure to switch to organic. But the use of biofertilizer organisms that enrich the nutrient content of the soil. The main source of biofertilizer, bacteria, fungi, and cyanobacteria nodules on the roots of leguminous plants formed by symbiotic association of rhizobium. This bacteria fix atmospheric nitrogen in the organic which is used by plant as nutrient. Other bacteria can fix atmospheric nitrogen while free living in the soil. Example, Azospirillum azobacter, thus enriching the nutrient content of the soil. Fungi are known to form symbiotic association with plants mycorrhiza. Many members of the genus Gromospore mycorrhiza, the fungal symbiont in this association absorb phosphorus from soil and passes it to plants having such associations show other benefits such as resistance to root-borne pathogens, tolerance to salinity and drought, and an overall increase in plant growth and development. Cyanobacteria are autotrophic microbes widely distributed in aquatic and terrestrial environments, many of which can fix atmospheric nitrogen and abandon nostoc oscillarian benefits. Cyanobacteria serve as an important biofertilizer. Blue-green algae also add organic matter to the soil and increase its fertility. Certainly, in our country, a number of biofertilizers are available commercially in the market, and farmers use these regularly in their field to replenish soil nutrients and to reduce dependence on chemical fertilizers. AIDS is treated by enzyme-like immunosorbent. Human Genome Project. The sequence of the bases in the DNA that determines the genetic information of the organism. In other words, genetic makeup of an organism and individual lives in the DNA sequence. If two individuals differ, and the DNA sequence should also be different at least at some places. This assumption led to the quest of the finding out the complete DNA sequence of human genome. With the establishment of genetic engineering technique, where it was possible to isolate and clone any piece of DNA and availability of space and fast techniques for determining DNA sequences. A very ambitious project of sequencing human genome was launched in the year 1990. Human Genome Project was called the Mega Project. You can imagine the magnitude and requirements for the project if you simply determine the aims of the project as follows. Human genome is said to be approximately 3 into 10 to the power 9 base pair, and if the cost of the sequencing required is US dollar 3 per BP, the estimated cost in the beginning, the total estimated cost of the project would be approximately 9 billion US dollars. Further, if the obtained sequences were to be stored in the type formed in books, and if each page of the book contained 1,000 letters and each book contained 1,000 pages, then the 300 such books would be required to store the information records from a single human cell. The enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and analysis. HGP was closely associated with the rapid development of a new area in biological bioinformatics. Goals of HGP. Some of the important goals of HGP were as follows. Identify all the approximately 20,000 to 25,000 genes in human DNA. Determine the sequence of the 3 billion chemical base pairs that make up the human DNA. Store this information in databases. Improve tools for data analysis. Transfer related technologies to other sectors such as industry. Address the ethical, legal, and social issues that may arise from this project. The Human Genome Project has the 13-year project coordinated by the Department of Energy and the National Institute of Health during the early years of the AGP. The Wellcome Trust UK became a major partner, and additional contributions come from Japan, France, Germany, China, and others. This project was completed in 2003. Knowledge about the effects of the DNA variation among individuals can lead to the revolutionary new ways to diagnose, treat, and someday prevent the thousands of disorders that affect the human being. Besides providing clues to understanding human biology, learning about non-human organism DNA sequence can lead to an understanding of their natural capability that can be applied towards solving challenges in healthcare, agriculture, energy production, and environmental remediation. Many non-human model organisms such as bacteria Canaro habitis elegans, a free-living, non-pathogenic tomato, Drosophila, the fruit fly, plants, rice, and Arabidopsis, etc. have also been sequenced methodology. This method involves two major sequences. One approach focused on identifying all the genes that are expressed as RNA, referred to as express sequence tag. The other to the blind approach of simply sequencing the whole set of genome that contain all the coding and non-coding sequence, and the latter assigning different regions in the sequence will function. A new referred to as sequence annotation for sequences. The total DNA from a cell is isolated and converted into random fragments of relatively smaller size. Called DNA is a very long polymer, and there are technical limitation in sequencing very long pieces of DNA and clone is suitable host using specialized vectors. The cloning resulted into application of each piece of DNA fragment so that it subsequently could be sequenced with ease. The commonly used hosts were bacteria and yeast, and the vectors were called as bacterial artificial chromosome and yeast artificial chromosome. Fragments were sequenced using annotated DNA sequencer that work on the principle of the method developed by Francis Sanger. Remember, Sanger is also credited for developing method for determination of amino acid sequences in protein. These sequences would arrange based on some overlapping regions present in them. This required generation overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. These sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed only in May 2006. This was the last of the 24 human chromosomes, 22 autosomes, and X and Y to be sequenced. Another challenging task was assigning the genetic and physical maps on the genome. This was generated using information on polymorphism of restriction in the nucleus recognition sites. Some repetitive DNA sequence known as microsatellite, one of the application of polymorphism in repetitive DNA sequence shall be explained in next section of DNA fingerprinting. Some of the salient observations that run from the human genome products are as follows. The human genome contains 3164.7 million base pair. The average gene consists of 3000 bases per size, vary greatly, with the largest non-human gene being dystrophin at 2.4 million bases. The total number of genes is estimated at 30,000, much lower than previous estimate of 80,000 to 1,40,000 genes. Almost 199.9% nucleotide bases are exactly the same in all people. The function were unknown for over 50% of the discovered genes. Less than 2% of the genome codes for protein. Repeated sequences make up very large portion of the human genome. Repeated sequences are structures of DNA sequences that are many times, sometimes 100 to 1000 times. They are thought to be have no direct function, but they shed light one chromosome struck dynamic and evolution. Chromosome 1 has most 2968 and Y has the fewest. Scientists have identified about 4 million locations where single have based differential SNPS single nucleotide polymorphism pronounced substance occur in human. This information to recognize the process of finding chromosomal location for disease associated sequence and tracing human history.